Good afternoon. You know, for 29 years of this North American CF meeting, I've had the great opportunity to stand in front here and look with awe at an amazing community that is bonded by one passion: finding a cure and control for cystic fibrosis and ways to improve the quality of life and the length of life for every individual with this disease. You know, there is no community like this in the world, a worldwide community whose passion it is to make a difference in the lives of people with CF. You know, for the last several decades, at my age you speak in decades, I've had it by my side up here, Cam McLeod. And Cam sends her regrets that she cannot be with us today, but she asked me to read a letter to you from her. And she says to all of you, "I am honored to be able to send my best wishes to this remarkable team of talented researchers, caregivers, and friends at a time of great progress. And I hope that each and every one of you has a stimulating and enriching conference. Because of you, my son, Cam's son, and all people with CF have more hope and have a greater chance to live fulfilling and rich lives and to make their unique contributions to the world." Unique contributions to the world. Thank you all for what you do. My best wishes to all, Cam McLeod. Thank you, Cam. I think Cam is probably online. You know, as many of you know, last week I stepped down as president and CEO of the Cystic Fibrosis after 21 years at the helm, 35 years at the Cystic Fibrosis Foundation, and nearly 4 decades, I mentioned decades again, in the field of cystic fibrosis. I feel very fortunate to have been part of this community for these years. You know, one of the greatest and most inspiring jobs and parts of my job has been at this meeting. You know, we've been so fortunate to engage the best and the brightest, the most dedicated, the most passionate into the field of cystic fibrosis. And it has always been fun since our first meeting when we only had 440 people. This year we're going to have over 4,300 people at this meeting. And you know, we could fit in a room at about 1/10 of this size. So congratulations to all of you for making that happen. And when I speak of a worldwide community, I want to make sure you understand we have people here from 42 countries from around the world. That is amazing. What a community. You know, I've treasured the opportunities to meet with you, to opportunities to learn from you, and I've witnessed firsthand what your passion and your commitment and dedication can mean to people with cystic fibrosis. I think over the next couple of days, you're going to see firsthand the results of all you have done and know in the future what you will be able to complete. Thank you all so much. So whether you're working in a care center on the front lines of this disease, or whether you're toiling at a laboratory bench with the ups and downs of research, whether you're carrying out clinical research or you're helping to raise the funds that are so critical for us to accomplish your mission, you are a unique community. You are a unique community. And whether you're new to CF, it's great to see some people that have just new fellows, new postdocs, new caregivers here, or whether you've been involved in this disease for 4 decades or decades or more, you know, you are changing the face of this disease. And I thank each of you for your hard work and dedication for advancing our mission. You know, I call this the ultimate dream team. The ultimate dream team. Each of you has an assignment. You know what you got to do. But it takes resources. It takes fuel. It takes passion. And we are very fortunate in cystic fibrosis and this community is to have passionate volunteers who are out there raising dollars 365 days a year, rain or shine, asking their friends, asking their businesses, asking others to contribute to our cause. You know, we need that fuel to be able to fuel the progress and to provide the resources to the talented people that we've been able to bring to this disease. You know, I have had an opportunity to meet so many of these people, and I want you to meet some of these people this afternoon. And we have some volunteer leaders here from the state of Arizona, from both our Phoenix and Tucson chapters. And we have other volunteers here today from the west, mostly from the West Coast, but I've seen some people from the East Coast here. And I want to ask all of them to stand up, turn around, and see the people here who are committed to their efforts. And I want all of you to applaud each other for what you're doing to make this disease different. So please, all of the volunteers stand up. You know, I could go through a litany of statistics, and I may in a few minutes, but what you are doing is changing the lives of individuals with cystic fibrosis. It's transformational. And we want you to see a video clip, some people whose lives you have transformed. So if we could have the first video clip, please.
Advancements in CF research have led me to a better life, and that there are now so many companies competing with each other to come out with all these treatments and drugs, and I take a lot of these drugs. I take Pulmozyme, I take hypertonic saline. All these things are making our lives more quality, not just quantity, but quality. I'm living a better life, not just a longer one, and that's important to me.
The advancements that Cystic Fibrosis Foundation has made gave me a life. It gave me a life. I haven't been in the hospital in 5 or 6 years. I take care of myself every day. The medicines have really helped me breathe. Like, I can't imagine the hours, like, you know, the doctor's wives sitting, or their husbands sitting at home, you know, eating dinner by themselves because they're so enthralled in this. And it's just, it's amazing to see a community, to be a part of a community like this.
There's new ways to treat CF. You know, the vest, and now you can get a vest that you don't even have to sit on the couch. You can walk around with it. There's just so many different things that are constantly coming up, and it's so motivating and so encouraging.
I can link back just about every medicine and treatment and therapy that I do back to some development and some research that was done. So I'm really thankful for that.
To look at how advancements in general have affected my life, you have to go back even before that, because I mean, when they came out with Pulmozyme, that was a big deal, and that was before my time. For a while, I just took it for granted because it was just another medication, but as I got older, I learned more about what it does and what a difference it makes. They change your life. They change your chances.
If somebody's diagnosed today, like, that kid is fully expected to grow up and have a healthy life.
That's what it's all about, folks. That's what it's all about. You know, this has been another incredible year since we were together last year. You know, in July, we celebrated the Food and Drug Administration approval of the second drug oral drug to treat the basic defect in cystic fibrosis, or COMBI. It was approved to treat nearly 50% of our patients who have, or individuals with cystic fibrosis who have 2 copies of the Delta 508 mutation. This is the second drug that has been approved, and obviously we have the vast majority of our patients have the opportunity to participate and to be able to utilize these drugs that are being able to treat the basic defect. The exciting thing for us at this point is that we have, at this point in the United States, and we expect this year to have 23 new clinical trials underway for not only these drugs, but for other drugs that can have a profound impact on the quality of life for CF patients. Thank you all for helping to make this possible. You know, so I will have to share some statistics with you because they are very special for us. In the 60th year of the Cystic Fibrosis Foundation, since it was founded in 1955. But this year, for the first time, we have over half of our people with cystic fibrosis over 18 years of age. You know, median life expectancy is now over 40 years of age, but there's no one in this room who will accept either of those statistics, because that is just a point in time till we ultimately achieve our mission. You know, you have helped us reach these kinds of goals, interim goals, and the future is unimaginable for people with cystic fibrosis. The other thing that you probably have heard about is that we had a monetization in cystic fibrosis where we put significant dollars in the bank as a result of our early investments with venture philanthropy that brought these drugs to the marketplace. The exciting thing for us is that we're already using and investing these dollars to extend our ability to treat this patient on a daily basis, but to think of monthly therapies, yearly therapies, and lifetime therapies. We're already investing these dollars to expand our research programs, to be able to invest in the science of the future, to be able to ultimately cure this disease. We're investing in our care programs to make sure that the resources that our caregivers need will be available to them to help to deliver the finest quality of care to people with cystic fibrosis as possible. We're going to continue to expand the training programs for all people, not just the physicians, not just the nurses, but to our entire team that will take care of our patients, and to the postdoctoral fellows and the Schwachman awardees. That's what we want to do with these kinds of dollars. And we're also committed to assuring that the people with cystic fibrosis are not only going to have the opportunities to live more tomorrows, but to give them the opportunity to live better todays. We are expanding our efforts in our patient assistance resource centers, making sure that our patients have the opportunity to be able to navigate through the complex reimbursement communities. We're going to up our efforts to make sure that our activities on the Hill and now in the States are going to be assured that our patients have access to these new drugs. And one of the things I'm most excited about, and the fact that we have so many people online today with cystic fibrosis, is the fact that we are incorporating more adult activities into the efforts of the CF Foundation. This was not always possible for us, but now with more than 50% of our people over 18 years of age, we want to engage these adults in the decisions that we are making that can profoundly impact on the quality of life that we want all of them to have. So we have an incredible momentum. You know, this train is moving very fast. I'm sure it's not fast enough for the parents and the grandparents and the aunts and uncles that are in front of the room here, but it is a momentum, something that we only dreamed about a few years ago, And the fact that we're treating the basic defect now at this point, which was a dream that we really started to exploit and explore in the nearly 30 years ago, is starting to pay off for us. You know, over the years, I've had opportunity to get up here and share some the highs and lows of what we've done, and hope that we have and know that we have the passion to make sure that we can work through those highs and lows. There are twists and turns when you're looking to find a cure for a genetic disease. But one of the great pleasures I've had is the opportunity to read to you letters that we have received from parents, from individuals with cystic fibrosis, who really summarize the impact of what you are doing every single day. A few years ago, you may recall, I read to you a letter from a mother whose daughter, a family who had actually moved from the West Coast to the East Coast with the hope and the prayers that they might be able to participate in the first Kaleido trials. If you recall, this woman thought she had won the golden ticket to her daughter's future. And she thought she had won it when she was informed that her daughter was indeed going to participate in the first VX 770 trials. Today I'm going to share with you another letter. If you recall, I just said a few minutes ago that this foundation is 60 years old today, this year. And it was founded by a group of families who had been told that their children were not going to live to school age. That was really unacceptable to those groups of families. And they formed the nucleus of the Cystic Fibrosis Foundation 60 years ago. And from that nucleus has grown the Cystic Fibrosis Foundation, its care center programs, its research programs, and the kinds of statistics that I shared with you today. You know, one of the families there was the Tolson family, Doris Tolson. And many of you have met her. She was at many of these meetings in the early years. And at that time, her daughter was 2 years old. And she was one of those that this was totally unacceptable to. And for that, she enlisted the passion of her friends, other families, to create the CF Foundation. So the other day, I received a letter from Doris's daughter, Annie Cates, who is 62 years of age. And I want to share this letter with you because it came at a very special time for me, and I hope it will be a very special thing for you. It says, "Dear Bob, this morning I had the privilege of walking my 5-year-old grandson until his first day of kindergarten with his mother, twin 2-year-old sister, and his other grandma. For everyone, it was indeed a momentous occasion. For me, I was happy for Hunter, but I was also thinking about how miraculous it was for a 62-year-old with cystic fibrosis. I was walking my eldest of 4 grandchildren into kindergarten and feeling better than ever. Who would have thought that me, Annie Tolson Cates, who was originally given a year at best to live in 1953, would be celebrating the marriage of her daughter when I was at 58 years of age, and now at 62 years walking my grandson to school with a new med available to me that changes every cell in my body. I often think of the younger patients whose young lives will be dramatically impacted by Ocambi and Kaleido, and other drugs that will follow as a result of the efforts of the CF Foundation. Perhaps they will never develop diabetes, as their pancreatic ducts will not be blocked. Women will get pregnant much easier and bear children. And everyone's quality of life will be improved as they will have less respiratory exacerbations and fewer hospitalizations. These new drugs will clearly make this disease a condition that is manageable. I began Orkambi on July 22nd. So I am now, and at that point she was in her 7th week, as of this particular Wednesday. I am feeling so well. Since beginning my med, and very soon after, my coughing nearly ceased. I cough only occasionally, but my sputum is clear, and I have no viscous mucus to cough up. I've gained 4 pounds. This is magical, pure science fiction. My chloride channels are working. She asked me to do one thing here today. She says, please shout out my appreciation to the countless individuals, scientists, caregivers, her caregivers, who have refused to accept the status quo and believe that science, good clinical medicine, persistence, and perseverance will change the essence of this disease. Thanks to all of you, Annie Cates. To each of you, I want to say thank you for allowing me to be part of this amazing journey. It has been a journey in which I have met some amazing people. Most of all, the people with cystic fibrosis and their families. I have made many friends in this process, and each of you have had a profound impact on my life, my family's life, and what I think I have hopefully been able to contribute to the efforts of the CF Foundation. But like so many people that I know in this room who had thought they were going to retire, you can never leave this community. And I hear a few of them laughing in the audience here. You can't step off this train. You know, it's moving too fast. Even a PhD nerdy guy like myself would be stupid to try to step off this train. The momentum we have achieved at this community will not allow that. But I'm going to take off some time. I'm going to enjoy my wife. I think she might be, if she got through the traffic in DC, looking online. Mimi, it will not be in a Winnebago. I'm going to enjoy my family and my grandchildren. I'm going to be one of those obnoxious grandparents on the sideline telling my grandchildren, you miss 100% of the shots you don't take. And I think that's important. I'm going to give my wonderful executive assistant, but more important friend, Deanne Greer, who is up here, who's been an amazing partner for me in my efforts, a break. And I'm going to come back. I've got a few mendings I have to take of the wars that I've been involved with over the years, not many. But you know, I will be back, and I'm going to continue to be part of you until we ultimately cross the finish line. But my decision to step down was made a lot easier because I knew who would follow me. Over the years, I've made some good decisions. But one of the best decisions I ever made was to recruit Preston Campbell to work with me 17 years ago. As our Executive Vice President for Medical Affairs, he has spearheaded the growth of our Care Center program, along with Bruce, our patient registry. He has learned the science. You know, I think he now could be a medicinal chemist. He knows structural biology. He knows so many things. But what he also knows is that at one time, when he was at the bedside taking care of patients and visiting with their families, the only thing that they could hope for and the only thing that he could give them was hope. But as a result of his tenacious, his persistence, and his passion, and the support of the team that he's been able to put together at the CF Foundation, he has turned that hope into a reality. So today I'm proud to introduce to you Preston Campbell, President and CEO of the Cystic Fibrosis Foundation. Thank you all very much.
Mimi, I hope you're on. And Bob, thank you for your kind words and, more importantly, for all your countless contributions to the community of cystic fibrosis. And we're in your debt always for your outstanding leadership. And actually, I'm honored to be able to continue the great work that you started on behalf of all individuals with cystic fibrosis. Now, I'm going to come back to you in a minute, and I got to ask Chris for a favor here. He's been prone to slip away when he thinks we're going to honor him. So will you tackle him if he tries to do that? So there's a little drama here. We know that Bob may try to slip away. But I want to say it's great seeing you guys here in Phoenix. Beautiful city. I want to welcome the folks that are online. I think there are 400 online. Welcome to all of you. Mimi, again, I hope you're on, and I also hope that Annie's on. I hope that she's joining us as well. And Annie, congratulations, and Doris as well. This meeting, bringing all of you all together, is a highlight of the year for us. And the output of this meeting and your collective work over the years has really resulted in cystic fibrosis being the most amazing story in medicine today. And more importantly than the story, you've made a dramatic difference in the lives of individuals with cystic fibrosis and are paving the way for even greater successes. We've come a long way, and you all should be incredibly proud of the progress that we've made, the contributions that you've had. But we also know that our work is far from being done. If you take the example of CFTR modulators, yes, we've gone from 5% of patients being treated with CFTR modulators in 2012 with the Orkambi approval this year, almost 60% now. And that's incredible, but we cannot stop, we will not stop until we have this type of benefit and even more for 100% of all of our patients with cystic fibrosis. And if any of you are saying, wait a minute, that sounds impossible to me, Bob will be the first one to tell you that when we started high throughput screening to develop these CFTR modulators, people were telling us this is impossible. And so we don't take that lightly, and we only use it as a motivation to go forward. But even though we'll move forward with CFTR modulators, we are still decades away from what we think is our ultimate goal. And our ultimate goal is a one-time cure for cystic fibrosis, where individuals no longer have cystic fibrosis. And in the meantime, we'll focus not only on CFTR modulators, but also all the other therapies, treatments and assists that individuals with cystic fibrosis need to enjoy the best quality of life they possibly can today. And I actually, while I have focused on therapies, new drugs, etc., there are many other unmet needs for those of you that aren't focused on science and drug discovery that are needed. And that's why this conference is so important for all of you, and all of your contributions are so important. You, along with thousands of others around the world, are the global effort to cure this disease. And I just want to thank you, along with Bob, for being here, and thank you for making a difference. Now, I've stepped into a new role, and people ask me how it's going, and it's going well. It's going to go well, not because of me, but because of the great team that we have. And we will continue to pursue every new and exciting opportunity to advance our mission that we possibly can. And that work is actually going very well as well. And I'm happy to report that the medical programs are now being led by Bruce Marshall, Bill Skatch, and Michael Boyle. And I cannot tell you, we couldn't have a better team of individuals to do that. They're going absolutely crazy, though. Bill and his team, Chris, who's on that team, and Martin, Elizabeth, and Catherine, are creating and finding so many promising opportunities that the research funding is really increasing. But the good news for that is that the science has never been better and our therapeutics development pipeline more robust. In fact, to accommodate all of that work, the number of clinical trials this year has doubled over the number of trials last year. And Mike Boyle, who's in charge and making sure that all of those trials are done safely and efficiently, yesterday at the Therapeutics Development Network meeting was announcing new funding right and left. And Bruce Marshall, responding to our care centers being stretched so thin, has come armed to this meeting with his own announcements about funding. So what do I think about all of this craziness? Well, I think it's perfect. And I think it's perfect. Why? I think it's perfect because it's exactly what we need to be doing for people with cystic fibrosis to make their lives better. So I'm very thrilled with the work that crazy team is doing. I'll highlight, I have one shout out for one thing that Bruce is working on, and I've been really thrilled with an effort that they're doing. That there are different facets. I'll use care center model of the future, but I mention it because you may want to check this out, because they're also focused on how you can partner in the clinical care arena with patients in a better way. And I think that's really a robust thing to focus on. And they actually have a symposium tomorrow, Symposium 12, that I encourage you to go to. That's chaired by someone that I admire very much, Paul Batalden. Had the pleasure of working with Cindy George and Mary Lee Phillips, who's an outstanding person with CF who's led many efforts for us in the past. So tomorrow, Symposium 12, please check that out. And then Mary Dwight and her team are also going crazy. But then Mary is kind of like that. Anyway, but they're charged with making sure that the folks that we care for with CF have access to all these therapies that are life-changing. And I want to report that they have some really neat and exciting programs that they've developed that you can see if you go down to our booth, the CF Foundation booth, or over to the CF Foundation booth in the exhibit hall. Check that out. See the amazing work that Mary and her team have been doing, and see what's available to make our patients' lives better. Now, there's one more important initiative that I just want to mention, and that is that we want to listen to individuals with CF better. We want to learn from them, and we want to embed them into all that we do. In order to accomplish that, we're going to start a new division at the CF Foundation. We're going to create that to make sure that we maximize this opportunity. Partnership with those who are most affected by this disease makes perfect sense to me. And although this initiative is very early, I'm extremely excited about it. And the reason is that I think it's the key for helping people with CF live better lives today. You see, we want to enable people with CF to add tomorrows and a bunch of tomorrows, but we also want them to live better today. Now I'm going to go back to Bob, but Chris, thank you. You scared him. He didn't run off or anything like that, so thank you. But before I do, I really have one other shout out, and that is to the folks that I've been working with recently in the executive team, because they've, quite frankly, been incredible. In fact, the whole CF Foundation has been incredible. I just cannot recognize everyone. So let me focus on the executive group, and that begins with my assistant, as Bob mentioned, Deanne. I have to mention Jackie Thompson, who's been my partner, has taken care of me, and is the reason I'm where I am today. And she is, quite frankly, an angel. I also have to mention Vera Twigg, our CFO, who really keeps so many things running well at the CF Foundation. Is just a remarkable lady. We have two new positions, two new hires in the executive office. I hope you meet them this week while you're here. The first is Mary Beth McMahon, who's our Chief of Staff. She has come to us from our communications group, really making a difference in this new position. And the other is Leah Bloom, who is the Chief of Strategy. She came to us about 2 or 3 months ago, really helping us think strategically about how we're going to move all these initiatives forward. Both of them are making a huge difference already. And I also want to point out Christina Hess, who isn't with us here today, but also Deanna Greer, who's helping us move all of these initiatives together. And then I'm blessed to have a very fantastic board. And I'm sorry that Cam cannot be here, and I hope that she's watching. So, Cam, I want to send our best to you from everyone here and hope that you and Bob are doing well. And I just have to tell you that I'm a blessed man to have so many qualified individuals to work with to help us make a difference in the lives of individuals with cystic fibrosis. Now, Bob, as we look to the future, we are quick to realize that we could not have reached this amazing place in our history that you described, in some ways I described, and everyone in this room knows about, without your leadership. When we needed science, Bob committed $10 million to the research development programs to create the science, and out of that effort came the cloning of CFTR. When armed with the knowledge of the blueprint of CFTR and it wasn't being exploited into new therapies, Bob created the therapeutics development program. And out of that effort came 2 drugs that treated the basic defect, CFTR. Bob, you've had an amazing career. You've really changed the face of this disease. You're taking us from no hope to hope. And if you look at what's happened with the survival during Bob's tenure, it's gone from 18 to over 40 years of age. And if you look at the whole CF population, this is pretty cool. During his leadership, he's added hundreds of thousands of years of life to the individuals with cystic fibrosis in the United States. Now, I'd like to show a quick video about what some others think about you, Bob. So if we could run the video, check it out. How has Bob impacted the CF community? That's easy. We've gone from no hope to hope.
He cares so deeply about patients with CF and their families.
He has inspired so many in the medical and scientific community.
I was one of the very fortunate people that hired Bob away from the NIH. And I don't think they ever forgave me for doing that. But I knew he was right for us, and he went on from there and made great things happen for us.
Having him on our team means that we have changed the course of this disease.
We can now think of, for the first time in our lives, of going back and identifying the gene that causes cystic fibrosis.
U.S. and Canadian researchers today announced a breakthrough which could lead to treatment or a cure for cystic fibrosis.
When he came, we really had nothing. And armed with $400,000, he committed $10 million to start the research development program, which 5 and 9 years later gave us the basic defect for CF and the genetic code for CF, which has enabled really everything that we've done since.
His real focus was the cure and bringing new drugs to market. But he didn't forget that people with CF have difficulty today, the symptom burden, the treatment burden.
He understood that while you're developing basic science, you have to be thinking about how do you get it into human trials.
He has taken a disease for which there was no hope and now made it the model of how to take on and defeat a disease. And this is true not just for CF, but this is true for all of medicine.
When people ask me to provide an example of how patients, caregivers, researchers, a foundation, NIH, and industry can all work together to find cures, I point to cystic fibrosis.
And now, within the last year or so, the finding of Kalydeco and Orkambi are mind-blowing.
There is not a single day that goes by that I am not thankful that we have that medicine, because it truly has changed just everything about my son's life and our whole family.
The greatest contribution that Bob has made to CF research is probably the uncanny ability of finding a direction to go, being able to recruit experts in that field, and then guiding those experts towards a common solution that no one ever thought would be possible.
I think Bob gets his drive from the families and the patients, and knowing and seeing that all the hard work that we've put into this organization is paying off.
Bob's greatest contribution has been the culture of collaboration and hope that he has created in the CF community.
Bob really makes me feel hopeful, not for the fact that, I'm hoping someday a therapy may be created. It's hopeful in the fact that I know it's going to happen. It's just a matter of when.
It's hard to describe him in one word.
Go-getter.
Compassion.
Inspirational leader.
I would call him brilliant.
Driven and focused.
Passionate.
Truly remarkable in all aspects.
The only thing that I would say to him is thank you. I'm able to wake up every morning and live a life that I've only been able to dream of.
If you notice, he's already telling me what to do. And the darndest thing happened to me. I stepped down just last Wednesday and I had to take a Greyhound bus out here. So already, anyway, Preston.
So no, don't go. So
what are you gonna do, Preston? I have no idea.
First of all, this is not a plaque. Bob hates plaques. This is a beautiful silver tray, and it says in honor of outstanding service, visionary leadership, and life-saving progress towards a cure, October 2015. Bob, I can speak for everyone. Thank you. We're glad that you're not jumping off, and we love you. And we thank you that you'll be following us. So
okay, thank you, buddy.
Okay.
As I said, and I cannot say anything more than thank you all for allowing me to be part of this journey with each and every one of you. We are making a difference. The best is yet to come. The best is yet to come. And I have never been so optimistic about the kind of energy, the passion. When you bring the right people together, you give them a great purpose for finding a cure, you combine that with passion, and you have the secret sauce for our future success. Thank you for allowing me to be part of it. Thank you so much.
2 quick awards. So we have one more way to honor Bob, and I'm delighted to share that in Bob's honor, the CF Foundation has created the Robert J. Bell Therapeutics Development Award to recognize members of the CF scientific community whose work embodies and carries on Bob's innovative, determined, and collaborative spirit in driving advances in CF research. It's my privilege to present the inaugural Robert J. Bell Therapeutics Development Award to 2 groups of incredibly talented individuals whose work has transformed the field of CF drug development. First, I'd like to recognize 2 leading researchers who were instrumental in the early efforts to adapt high-throughput screening to cystic fibrosis and have brought many other useful tools to the community. The first is Dr. Alan Berkman, a professor at medicine and physiology. You can clap for Alan.
Come on, Alan.
Alan is a professor at the University of California, San Francisco School of Medicine. He became the director of the CF Research Development Program there in 1991, a position he holds today. He's been the senior author on numerous articles on CFTR modulation, both potentiators and correctors, and he early on championed the idea of high-throughput screening for cystic fibrosis. An individual that he worked with, also receiving the award today, is Dr. Louis Gallietta, also known as Gino by his friends. Gino is a principal investigator at the Gaslini Institute in Genoa, one of the largest pediatric hospitals and research institutes in Italy. He's internationally recognized for his groundbreaking research in the functional and molecular characterization of ion channels and epithelial cells, as well as for his work developing new fluorescent proteins in the study of channels and identifying CFTR pharmacologic modulators. So I'm very proud to present the first Robert J. Bell Therapeutics Development Award to Drs. Berkman and Gallietta, pioneers in the development of high-throughput screening and true partners in this effort. Unfortunately, Alan is traveling and couldn't be with us today, but please join me in welcoming Gino to the stage to accept this award.
So I'm very honored for this. I thank Bob, Preston, Chris, and the whole foundation for their support. Thank you.
Thank you. Thank you,
So we're sorry Alan's not here. It's also my privilege today to recognize the research teams at Aurora Biosciences and Vertex Pharmaceuticals who took on our pursuit to treat the basic defect with innovation, energy, and a growing commitment to our mission. Vertex's huge and pioneering effort has yielded clinical results far beyond anything we ever imagined. And again, it would be impossible to recognize everybody who was involved in this effort. There was an army of folks involved at both Aurora and ultimately Vertex. But as a start, I'm proud to present the Robert J. Bell Therapeutics Development Award to the 19 scientists whose names are listed on the screen. Their early efforts led to the clinical proof of concept that the small molecule approach would ultimately help bring CFTR modulators to fruition. So please join me in welcoming Paul Negulescu, a Vice President of Research at Vertex and member of the initial team at Aurora, to accept this award on behalf of his team.
Hey, buddy. Good morning. Thanks for answering the phone. Thank you.
I've been waiting 20 years for that. Wow, this is incredible. Just like Gino said, this is quite an honor. When we started this program 15 years ago, it was unprecedented, it was bold, it was big, and it was all driven by Bob's vision for the future and his faith in science. And on behalf of the Vertex research team, both past, and these were the folks involved in our first paper describing our high throughput efforts, but also present and future, I'd like to thank the foundation, thank the CF community, the advisors who gave us so much good foundation to build our efforts on along the way. It's a true privilege to be up here and to be part of this community. It's also a special honor to get the first inaugural Bob Bell Award. Bob was the catalyst for getting us on our CF journey, and he was the person who along the way kept us focused on the science and kept us focused on the patients and taught us what it is to be part of the CF community. And I have to say that the stare that he gave us when we weren't working fast enough was also quite motivating. So just want to thank Bob for that. You know, as all great leaders, he brought out the best in us, and I know we're not the only ones. So thank you, Bob. Thank you to the CF community.
Great team. Congratulations to you both. This is a great honor, and we thank you for your contributions to the CF community, and we'll make sure that Alan gets his award as well. Now it's my pleasure to introduce Dr. Chris Penland, who has a few things to say, and will introduce our plenary speaker today. And we're fine on time. For those of you that are worried about time, we're absolutely fine on time.
He hasn't cut out the cocktail party.
So thank you, Preston, and welcome to everybody to fall in Phoenix, where just last week it was 170 degrees. Now it almost feels like Christmas outside, I think. I want to reiterate Bob's and Preston's welcome to all of those in attendance and those joining us online. Before I introduce today's plenary speaker, I want to recognize two other groups. So annually, this meeting is designed to recognize your most recent accomplishments, but more importantly, to accelerate our community's progress. In meeting that goal, I want to thank Ms. Amanda Leonard and Dr. Wayne Morgan, the conference co-chairs, for their leadership. They and the members of the conference planning committee have been working hard for almost a year to push our boundaries of knowledge regarding CF. So please join me in giving them a round of applause. Another group deserving of recognition are the conference's sponsors and exhibitors that help to improve the quality of this meeting and are also committed to the CF mission. They bring important technology and expertise to our effort, so please stop by their booths, learn what they're doing, and thank them for their commitment and support. So lastly, it's my great pleasure to have the opportunity to introduce today's plenary speaker. Dr. John Paul Clancy, or as many of you know him, JP, was born and raised in Iowa, where he completed his undergraduate and medical degrees at the University of Iowa. While a Hawkeye, he had the opportunity to train in the laboratory of Michael Welsh. This seemed to be a good choice, as JP's first of 7 publications as a medical student was in the journal Science. After a pediatric internship and residency at the University of Virginia, JP was recruited to the University of Alabama, Birmingham, where he trained with Eric Sorcher, eventually joining UAB's faculty in 1996. During his time at UAB, Dr. Clancy was a Leroy Matthews Physician Scientist Awardee, served as Associate Director of the CF Care Center, directed the Pediatric Pulmonary Division, trained or mentored 24 fellows and PhD candidates, and many more residents and master's candidates, where he eventually rose to the rank of professor. In 2011, Dr. Clancy was recruited to the University of Cincinnati and Cincinnati Children's Hospital, where he was the inaugural Tom Boat Endowed Chair in Clinical and Translational Research, and is now the Gunnar Esiason Cincinnati Bell Chair of Life Sciences Research. Dr. Clancy, with AP Nairn, also co-directs the Cincinnati Children's Hospital CF Research and Development Program, and we will shortly hear some of the work they are now focused upon in the laboratory. JP has published more than 100 scientific articles, served on numerous organizing and review committees for the CF Foundation and other organizations, chaired or been a member of numerous data safety monitoring boards for NIH-funded projects, advised numerous companies in their therapeutics development efforts, and served as site PI on a large number of multi-center clinical trials and lead investigator on 3 national clinical trials. Now, in his free time, JP is a husband and father of 3, but he still found time to appear on NBC's Today Show, which perhaps kindled his fondness for public performance. Please join me in welcoming Dr. John Paul Clancy to the stage.
A beautiful bunch, a ripe banana. Hide the deadly black tarantula.
Please join me in welcoming JP to the stage.
I'm going to kill her. Her name is Kim, if anybody wanted to know, by the way. But anyway, thank you so much. What a wonderful tribute. What a great opportunity. What a great meeting. What a big room. And I'm really thrilled to be able to lead off the talk today. And I was just thinking to myself, sitting down there, what do you say after an icon like Bob Bell is announcing his stepping down, moving on to different projects? The one thing that came to mind, Bob? No pressure. So thank you again. And today I'm going to be spending time talking about what I think we're moving towards in the care of CF for the next century, personalized medicine, and I look forward to updating you on what work is going on and where I think we'll be in the next coming years. Quick summary of disclosures. Nothing that I'll be talking about today should be off-label. So I also wanted to mention that I had the opportunity to do this talk in 2006, And as I was thinking through, how do I open this talk and so forth, it was a big time for me. My second daughter was born. That's Maeve. And I remember I flew out 2 days after she was born, gave a talk, flew home. And of course, being ill-prepared, I used, shameless pictures of my children to win the crowd over. And hopefully that worked. But I wanted to give you an update. So now that we're, 9 years later, she rocks the Jackie O look, and if you could look carefully, she reads about snakes, and if you have any questions about snakes in southeastern Ohio, I can help them be answered. But it also gave me an opportunity to think about what has changed since that time, and sometimes those of us who are taking care of patients, doing research, doing clinical trials, we're in the weeds, and this gave me a moment just to step back and say, in the last 9 years, what's different? We have new standard therapies. We have pancreatic enzymes that are finally approved, new antibiotics, new mucolytics. We have genotype-directed therapies, modulators and others. We have mutation-agnostic studies that are going on. We have patient-derived model systems and biomarkers in development. And we've got new novel study designs and monitoring tools. And the point is I'll be talking about these today. And when I step back, it just reminds me that we have made amazing progress in a short time, just in my second daughter's time here. So I'm really breaking this talk into 3 parts. The first is talking about the concept of personalized medicine, what it is, and how it's being considered both on the national stage and also in the CF community. Talk a little bit about therapies that are driven by genotype and mutation, also by responses, or neither of those, and we'll get into that in just a few minutes. And then finally, how we're trying to bring some of these concepts into a personalized approach to CF. And this includes looking at personalized model systems and disease monitoring that.
If we start today and seize this moment and the focus and the energy and the resources that it demands, there is no telling how many lives we could change. And every single one of those lives matter.
He's pretty good, isn't he? Yeah, all right. And that was also in part discussing some of the advancements specific to cystic fibrosis and new therapies. And soon after that, there was a publication in the New England Journal of Medicine by Francis Collins and Harold Varmus that really described this new initiative in precision medicine. And I'll just read this, just bear with me for a moment. The initiative will encourage and support the next generation of scientists to develop creative new approaches for detecting, measuring, and analyzing a wide range of biomedical information, including molecular, genomic, cellular, clinical, behavioral, physiological, and environmental parameters. It will also pioneer new models for doing science that emphasize engaged participants and open and responsible data sharing. And those are really huge, big, ambitious ideas. But when I look at that and see what's happening in the cystic fibrosis space, I feel confident that we are well on our way to achieving these goals. But I've kind of been wrestling with the term personalized versus precise. And so being a very, 21st century guy, you could tell by my dance moves, obviously, I chose to go to Wikipedia and sort of get my hands around what those two things, really what the definitions are. And you can obviously find a lot of them. But personal or personalized is to design or tailor to meet an individual's specifications, needs, or preferences. And precise or precision really focuses more on accuracy and exactness. And so, which one? I mean, I kind of hear them back and forth. I think what's exciting is we have the opportunity in CF. We are practicing medicine, I think, in a very personalized way now. We just now have information and tools that are going to let us do this in a really precise fashion. And I think that, to me, summarizes it best. So those of us who take care of CF patients, we do personal care every day. Personalized care is part of our culture. We do culture-based treatment of pulmonary infections. Not everybody's on an inhaled antibiotic all the time. We do enzyme replacements based on symptoms and feedback, vitamins and nutritional supplements that we use in different ways for different patients at different times. Insulin is used in a lot of different ways to treat growth failure, to address glucose intolerance, and obviously to manage CF-related diabetes. And obviously, if you want to get into a big debate with people in cystic fibrosis, choice of airway clearance techniques, length of antibiotics, duration, all those things are very personalized. And so, but I think the other aspect that I remember, and I'm thinking of Bob as I say this, is from their perspective, in patient's perspective, it's all personal. And we can be precise as we want, but if it isn't personal, we're not doing our jobs correctly. So this slide summarizes the what evidence-based medicine has done to outcomes and median survival of CF patients. It's being shown in a slightly different way. What we're really looking at, oh, and I'm sorry, got a little scrunch there on the side, but on the y-axis is age in years, and on the x-axis are like 5-year blocks moving 1 year ahead, so it kind of averages some information. And what we can see is with the initiation and the evidence-based application of a variety of therapies that we use every day in cystic fibrosis, there has been a steady improvement in median life expectancy and improvement in quality of life. But this is a slide I've shown before, and I'm going to probably keep showing because I keep adding to it. The challenge is all that exciting new therapies that we can give to patients is more therapies that we're giving to patients, more time. The average adult with CF to stay well spends approximately 2 hours a day staying on top of their cystic fibrosis. And so I think we're moving, maybe not immediately, but we're starting to think about how do we do this on a personal way. We can finally realize that one size doesn't fit all, and if we expect to be able to add and add and add, we're going to see diminishing returns, and I think we're finally coming to realize that. So before I get into some of the therapies that are in development and how we're going to try to tailor them to patient needs, I wanted to just give a quick summary of what I think most of us in the room are going to agree about CF and CFTR. We know this gene codes for a protein, and this protein helps move salt, and specifically it moves chloride and bicarbonate and regulates other transporters like the sodium transporter. And when it's not working, it creates a vicious cycle that is the source of symptoms in cystic fibrosis. Buildup of thick mucus, chronic infections, long-standing inflammation, and ultimately airway damage that eventually is unfortunately the cause of morbidity and mortality in the vast majority of CF patients. What's exciting is that each one of these is a target. CFTR modulators, concepts of gene transfer, gene and RNA editing really are targeting upstream causes of cystic fibrosis. Treating the thick mucus with hydrators and mucolytics really helps to clear mucus and lead to daily better respiratory symptoms. Antimicrobials deal with the infectious issues that are chronically part of cystic fibrosis. Persistent inflammation is tackled with anti-inflammatories. And so we should all feel really good about this broad pipeline that is targeting all these different aspects of cystic fibrosis. If you go off to the Cystic Fibrosis website, the North American CF Foundation, you will find this updated pipeline. I'm not exactly sure when I downloaded this, and I think the only message I'm trying to get to you is it's very broad. We have a lot of activity across a lot of different areas of cystic fibrosis. And what is a little scary is it's kind of broad, and how are we going to be able to tailor these therapies in the most efficacious way for our patients. So that leads me in sort of the next part of the talk, which is really addressing new therapies that are targeting patients based on the mutations they have, perhaps by the drug response that they have, or perhaps neither. And these include genotype, serotype, and genotype agnostic approaches to cystic fibrosis. So I think we realize that in cystic fibrosis there are thousands of mutations which cause disease. And people have wrestled about how should we categorize them, and I acknowledge that there's not a perfect system. A system that I think is a great starting point is really breaking them into different classes. These are non-exclusive, I think we've come to learn, but they do give us a starting point to understand how we can tackle different mutations that cause cystic fibrosis. Class 1 defects lead to problems with protein biosynthesis, and there's very little, if any, functional CFTR. Class 2 mutations cause problems with CFTR folding. Delta F508 is the most common cause of CF and is in this membership class. And then there are a variety of mutations which get to the cell membrane to where they're supposed to do their job, but they just are not doing it very well for different reasons. We can have a gating problem, which is called class 3, where the protein is basically at the membrane, but it's stuck closed, and it's not able to open. There can be mutations in which the pore, or basically the little path or channel for ions to go through, is too small, and it doesn't allow them through very well, and those are called conductance. And finally, there is a group of problems which lead to low levels of working CFTR that is hard to put into one category, but we tend to put them into this class 5 group. And when we look at this, each of these sort of have different strategies to help restore function. As we move from left to right, suppressors are drugs which we think will be valuable for patients who have premature termination codons. Potentiators and correctors appear to be very valuable for patients who have the Delta F508 mutation. Potentiators help to activate gating mutations and may be applicable to some of these other surface CFTR mutations. And finally, if we wanted to think about approaches that have nothing, that don't have to require a given mutation, gene transfer becomes an option, and also theoretically approaches such as DNA and RNA editing. So another grouping that we can go by that really focuses on the sort of the therapeutic pipeline that's ongoing right now can sort of break down coverage of CF patients by the types of therapies that are in clinical trials. We know that about half of patients with cystic fibrosis have 2 copies of the F508 mutation. About 40% have 1 copy of the F508 mutation. About 6 to 7% have a gating or R117H mutation. And there's a small group remaining in the 5 to 10% range, which don't fall well into any of those categories. And kind of sprinkled throughout that group of patients who don't have 2 copies of F508 are 5 to 10% of patients who have premature termination codons. And all of these are approaches, each of these sort of represents a group of patients who are being considered for drug development. So the landmark study from 2012 that looked at ivacaftor in patients with at least 1 copy of the G551D mutation is known as the STRIVE study. Again, kind of a medium-sized study in the CF space of 160 patients, and they have kind of moderate lung disease. And what we're looking at was 2 of the important clinical outcome measures from that study. On the left side is lung function, or FEV1. The right side is acute pulmonary exacerbations. And what we see is that patients treated with ivacaftor had a rapid, sustained improvement in lung function, 10 to 12 plus above baseline. That was very sustained throughout the course of the treatment. And then patients, their risk of pulmonary exacerbation was cut in half, even more like 60%. And this mutation was targeting one of the gating mutations in cystic fibrosis, and this again is the most common member of that class. And probably most importantly for all of us who are currently trying to bring these drugs forward today is this is a benchmark we can use. This really translates into clinical benefit. And when we can achieve this, we know we could be making levels of CFTR function that have really meaningful clinical impact. There have been additional potentiator successes that have recently been presented and or published. First is the Connection study, which looked at patients with non-G551D gating mutations, which includes at least 8 of those. While the study design was very different, it had a very similar kind of evidence of activity and led to FDA approval. The Kiwi study, which basically looked at patients with gating mutations in the ages of 2 to 5 years. And based on the success of that study, it led to FDA approval. An exciting finding in that study was that there may have been some evidence of improved pancreatic function, which would really be the first time that's been demonstrated. There are some safety issues we have to watch in the younger population, and obviously we're watching those carefully. And then there's the Conduct study in which the R117H CFTR mutation patients possessing that were treated with ivacaftor. And they also showed clinical benefit. And while the impact was a bit variable because there was a lot of variability in that disease, what's exciting is that the FDA looked at the data, heard from the patients, and determined that the presence of R117H was enough to lead to FDA approval. And so what this has led to is availability of this therapy to a new and growing group of CF patients. We also know that the most common cause of cystic fibrosis is the delta F or F508 mutation. That's a very, very different beast, I think most of us have come to realize. And this is just an example from a publication in the mid-2000s showing an airway cell with CFTR at the top in green. And that's really telling us where normal working wild-type CFTR belongs. It's up there, it's doing its job, it's moving salt, everything's working. And unfortunately, when patients have 2 copies of F508, it doesn't get there. The delta F508 mutation causes a problem with how the protein folds. It gets stuck in compartments inside the cell, and it's not able to go where it needs to and do its job. Now, correcting delta F508, that's a symposium in and of itself, and there are some exciting ones that I think that many of you will want to take in while you're here that really are getting at the underpinnings of the cause of the delta F508 mutation. But I'm just going to summarize it that it's complicated. There are at least 2 kind of general problems that need to be overcome, and possibly more. And this is a slide that is from an old plenary talk, but I think it kind of captures the idea pretty well. When a protein is being made, it needs to fold into a certain shape. And in CFTR, it has lots of little domains that makes it kind of complicated. The NBD1 domain is where delta F508 is, and when F508 is missing, it doesn't fold up right. It's kind of loosey-goosey, and that kind of gets in the way of another role in which that part is supposed to talk to another part. And when those communications don't happen, you end up with a protein that's unstable, and it's not able to go where it needs to go and do its job. And so what that tells me is that one drug is probably not going to get us all the way there, and I think we have come to know that. But what's been exciting, I think we can say, is that there was a recent press release about drugs that are really trying to tackle more than one folding step in the F508 that was released today by Vertex, and we're hopeful to see those studies beginning in the next handful of months. So this problem is starting to be addressed in the clinic. But there has been obviously the exciting release of the traffic and transport studies, which looked at the impact of ivacaftor and lumacaftor together as a co-therapy in patients who have 2 copies of the F508 mutation. And this is the largest undertaking of an interventional trial in the cystic fibrosis space. And if I'm wrong, correct me, but I'm pretty sure that's right. And what was shown was a significant improvement in lung function. What we're looking at is FEV1. The straight line at the bottom is the placebo group, and the green and the blue are patients who were on 2 different doses of ivacaftor lumacaftor, which had very similar effects in at both doses. And as importantly was the impact on acute pulmonary exacerbation shown here, in which the patients on drug in the blue and the green had a slower risk or a reduced risk of pulmonary exacerbations. And so that led to the approval of Orkambi that has now become available to our patients this summer. And I think many of us who are in the CF care space have been working very hard to get patients on drug over the last several months. There is also a very active program ongoing to bring drugs to patients with any F508 mutation, and this is the VX661 phase 3 program. Any of those participating in clinical trials here know how busy this is and how exciting this is. But basically what it does, it looks at patients who have at least, they have to look at it in different ways. As we move from left to right, you have patients with 2 copies of F508, 1 copy of F508 and a gating mutation, 1 F508 mutation and a functional mutation, and the 1 F508 and a non-functional mutation. And because of sort of different numbers and sizes of populations, you have to do very different studies. So what's exciting, though, is despite the need for different studies and different designs, we might have a common co-therapy in the not-too-distant future for anybody who has an F508 mutation. So if we go back and visit our breakdown of patients based on genotypes and drugs that are currently in development, we have therapies that are now available for the patients who have 2 copies of F508. We have therapies available for patients with gating and the R117H mutation. We have therapies under development for patients with the F508 as 1 copy. And there remains about 5%, and also, in addition to that, the patients who have PTCs, who are ongoing studies currently with Adalurin, which are currently in phase 3, and we're very excited to hear about those results. But there is a group, of patients who have rare mutations. These are mutations that just not well characterized, or not a lot of patients for us to really band together into any kind of a clinical trial. So how are we going to get drugs that really target their cause of CF to them? And that's really what we're going to be thinking about as we go forward. So what I've shown you up till now is thinking about developing therapies that is based on a genotype or a mutation type. And that's kind of what's shown here by my 5 little cartoon cells. But we can look at this in a very different way. We could actually take mutations and then take a mutation perhaps that's not as well defined and then test and see if it has a drug response. Is it possible that that therapy in this patient who has a not really clear-cut mechanism of mutation might actually just benefit if we knew how to measure it better? So how do we do that? Well, this is accomplished, at least in theory. One approach is by doing what are called N-of-1 trials. And in this type of study design, it's basically patients serving as their own controls. This is not new. This is an idea that's been around for a while, but it's really starting to be embraced by the CF community as a way to help, a pathway for us to help get patients matched up with drugs that work for them. And in this concept, patients are randomized often on a given treatment. There are periods where they have a number of different parameters monitored in a very precise sort of fashion while on and off drug. And it's hopeful that this type of approach is going to help us really bring some of these drugs to patients who have mutations who just unfortunately don't have other clear paths to get there. And what I understand from a recent press release is that ivacaftor has been submitted as a new drug, basically an application to use this data that was generated from this type of study to bring ivacaftor to therapies with partial function CF mutations. So we look forward to hearing about those results in the springtime about how we may be able to expand ivacaftor therapy beyond the current use. Finally, I wanted to mention mutation-agnostic therapies, and in this concept, we really don't have to worry too much about what's the cause of their CF, which type of mutation, because in theory, it's going to address all of them by basically replacing it or giving enough working CFTR to do the job. Now, the studies that have recently been conducted and reported have just looked at airway delivery, but there's been some exciting results that suggest that there may be a direction in which this type of approach could happen. In a phase 2b gene therapy study that was reported by Eric Alton and is also in one of the symposia today, I do believe, the study included the delivery of a lipid DNA conjugate in a breathing treatment once a month for a year compared with placebo. And what we're looking at is the impact of that treatment on lung function in blue and in the placebo in red. And obviously the effects are not dramatic, but what we really did see was a statistically significant difference. I don't think we're perhaps saying that this is the right therapy currently, but I think what it tells me is that for the first time we have a measurable improvement in lung function in patients treated with a gene therapy. And I think it opens up the door to future therapies and research that uses this general approach. So the third part of the talk, I wanted to discuss bringing personalized concepts to CF. So I've talked about genotype-based therapies and talking about theratypes and how we might match drugs to patients in principle. But let's take it a step further. So perhaps we can get information from patients that tells us whether drugs are the right choice for them. And do we have tools that are now available in the clinic and also at home that can inform us about whether these drugs are really benefiting? And I think it's because when I look at what's in clinical trials right now, this is not a completely, by any stretch of the imagination, a complete list, but it's a complicated world out there. We have a lot of therapies in development, and how are we going to get the right therapies to the right patients when we have these different options, both those with rare and with common mutations? And I think an example that helps bring it home to me was published in 2014 by Mike Boyle and colleagues. This was the ivacaftor-lumacaftor phase, or one of the ivacaftor-lumacaftor phase 2 studies in patients who had 2 copies of F508. And what you're looking at is basically the improvement in lung function during co-treatment. And each one of those lines is a patient. The purple lines are the patients on active drug. The red little lines or bars are patients who are on placebo. And the y-axis is basically the change in lung function. And what we see is that what's interesting is there are patients who don't really seem to benefit, at least based on this measurement. Maybe there are other ones that would. But while these guys clearly are getting a benefit, at least in terms of lung function. And so it's probably not surprising. Not everybody responds to every drug in the same way. I mean, I take Claritin, my wife takes Zyrtec. I mean, that's just kind of the way it works. So I don't know why, but I think the point is maybe we can take this a little deeper. So why does patient 1 respond and someone else? Well, I think what the point is, we have the potential to have tools that are going to predict who responds and who's going to respond best to which choice. We definitely want to understand the variability of responses so we can figure out if that's something that we can address. And we think that this type of approach of really understanding this is going to help us guide future clinical trials. But I do want to just raise the point that I think regulatory conversation is going to be really important as we do this. How is the information that we're able to gather during patient treatment that inform, or even from cells derived from patients that might inform our treatment choices, how is that going to be viewed by regulatory authorities if we want that to become a drug that we can prescribe? So these are all, I don't have all these answers, but I think this is where we're going to be going over the next few years. So when we think about personalized model systems, there are at least 3 ways that people at the moment are thinking, and there are studies ongoing now and many abstracts downstairs that are sort of covering this topic. First one I'll mention is organoids, which are basically small little balls of cells, basically, that were derived from GI tissue, and I'll show you some examples of those. Second are airway cells, and specifically brushed nose cells from patients. There's been some breakthroughs in how we can grow those cells, and perhaps now that becomes a testing ground. And then third are stem cells, and these are basically cells that are derived from patients that are then coaxed into being CF epithelia, and therefore are a place that we can test whether CFTR works. Intestinal organoids have been considered as an approach to guide CF therapy based on some recent work that was published in 2013 by Jeff Beekman and Deckers and Cleavers in the Netherlands. And what this concept is a small little intestinal biopsy, usually from the lower GI tract, is isolated. Cells are obtained, grown in a special way, and they form these little microscopic spheres or little balls of cells. And what we're looking at on the top left here is an example of those little spheres in green. The top is from a patient who does not have CF, it says wild type, and the bottom is a patient who has 2 copies of F508. And as you move from left to right, what you see is that the CFTR has been stimulated, and the wild type organoid swells. It gets bigger. It basically, and that represents CFTR secreting fluid into the inside. And that's shown on the right side of this slide by looking at the impact of a variety of different correctors on the organoids from patients who had delta F508 mutations. We see that there is swelling or an increase in the area of these little organoids over time, which is shown in on the x-axis, and we have different combinations of drugs showing that there actually appears to be synergy in this model system. The bottom is just follow-up studies looking a little more carefully at these. On the bottom left is just looking at the cells themselves, what's actually in an organoid, and the blue dots are basically the nuclei. The green is a surface marker of the cells called actin. The CFTR is in red, and what we see in the top figure is that CFTR is present, and down below it's absent in the patient with cystic fibrosis. And finally, on the far right, we're starting to think about how it differs based on the amount of CFTR function. There are basically organoid swelling studies in HC, which stands for healthy controls, which is very fast and robust. In the middle are patients who have mild or partial function mutations, and in the bottom are patients who have 2 non-functional mutations. And so this concept is really being thought about as a means to determine whether drugs might be of benefit to CF patients, and is being piloted by a number of groups. This was a video that was taken by in the lab right next to mine, basically, by A.P. Naran and Moon, who works with him. On the left is an example of an organoid that is swelling after it's been stimulated. That's from a person who does not have cystic fibrosis. On the right is basically the same thing, a little organoid that from a patient who has 2 copies of F508, and it's been stimulated and nothing happens. And basically how big it swells is kind of a relatively simple readout of whether CFTR is working. So how about nose? Well, if anybody knows me, I have no problem putting something up the nose. I've been doing nasal PDs and more things up noses than I care to remember for the last 20 years. Why would noses seem to make sense? Well, there's a track record, at least in principle. Turns out primary airway cells from the lung have served as a great way to develop drugs like CFTR modulators. All the drugs that have been approved, and many of the drugs that are currently in clinical trials, if not all of them, at some point have been studied in primary human airway cells from patients with different CF-causing mutations. And so there is a sense that this might be a reasonable starting ground to predict things that might be beneficial in patients. And so, but that's not easy. Going down and getting airway cells from a given patient is not a simple thing. And so, can we use cells from the nose, and can these really serve as a testing ground for CFTR and other types of drugs, CFTR active drugs and other drugs? And this might be relevant to patients who have rare mutations. This could be for patients who have very common mutations but lots of choices. But we are wrestling with some basic questions, and there's a lot of labs working together to try to solve this. How do we grow them? Do the nose cells act like the lung cells? Do they respond? And probably the most importantly, does how a nose cell predict how a patient is going to respond? So the short answer, we don't have that answer today. But the good news is we have rapid advancement in our understanding. And this is just an example of some cells that were brushed from a patient with 2 copies of the F508 mutation. They've been grown out in a sheet, and then from this type of growing sort of structure, we can measure how well CFTR works. The green is the outline of the cells, the red is I think that's the tubulin, and the purple is the mucus. So they have some of the things we're expecting to look at. And this is a picture that came from Cal Cotton, who's an expert in growing airway cells from the noses of people. And what we're looking at is CFTR function, and that's really what's shown here in the middle. Each one of these is called an Using chamber tracing, and basically it's a way to monitor whether CFTR is working. All these cells were derived from a patient with 2 copies of the F508 mutation. In the top, they've gone through a series of stimuli to activate CFTR, and in the middle, we see this little bump, which represents maybe a little hair of CFTR that's being detected. At the top tracing in the middle of that tracing. The middle tracing, which says VX809, is basically the same cells but treated with VX809 to correct F508. We see an improvement or a bump up, which represents activation of corrected F508, and then you add 770 on top of it and you get an additive effect, and then it blocks with a CFTR specific blocker. And then the bottom is a very similar experiment done with VX661. So the point is, you can see that this type of model system, at least in theory, is a way for us to determine whether drugs are active. And, maybe we can come up with easier ways to grow these cells. I will tell you, it takes a long time to go from a brush nasal cell and isolate it, to grow it, to get it onto a sort of this right surface, to be ready to study in these chambers can take, close to 2 months. Another approach which we're piloting, and we're among other groups who are doing the same thing, is looking, can you basically make these little nose cells work a little bit like these organoids? And what we're showing here are little nose cells, spheres basically, that at baseline and then after we've stimulated with a drug to activate CFTR, and they swell. And yes, you can take really cool pictures if you want to of them that show us outlines of the cells and give us an idea of the types of cells that are actually represented there. The last thing that I wanted to mention is, in addition to having these personalized systems that are derived from patients, we also need to have the backside covered. How are we going to monitor whether these drugs are having any kind of effect on patients? And there are a number of things ongoing right now that I think are going to really be helpful guides for those studies moving forward. First of all, there are observational studies ongoing right now that are looking at patients before and after they've started modulator therapy. One of these is Prospect and the other is Goal. We'll talk about those briefly. There's been some advancements over the last few years in how lungs are imaged. Looking at MRI, which has some potential advantages that are unique and may be valuable for future drug use. And then finally, the development of apps that we can use, the patients can be using to monitor and track therapies as we make interventions. So I wanted to summarize just briefly the Goal and Prospect studies, which people may or may not be aware of. But these are both observational studies that are looking at obtaining data and banking specimens of patients before and after starting drugs that restore CFTR function. Goal is really focused on people with gating mutations and R117H, and it's basically for patients who are prescribed ivacaftor, and that study is going on very well, and I know there's some data that's being presented. I think it was at a symposium today that sort of summarizes some of that recent work. The Prospect study, which is very active right now, is looking at patients who have 2 copies of F508 before and after they start Orkambi. And we're also looking at patients who have sort of variable levels of CFTR function based on their genotype, getting samples from them and measurements. So we're going to have sort of a sense of what those measurements look like in people without CF, with a little bit of working CFTR, and with no working CFTR. And when it's all done, we hope that we will have a number of biomarkers such as multiple breath washout and LCI, fractional excretion of nitric oxide, pH pill to measure intestinal pH, insulin measurements in patients during glucose tolerance testing, mucociliary clearance assays, and a very robust and full bank of specimens from patients that is connected to the CFF patient registry. So this work is ongoing right now, and it's going to really set the foundation for exciting work in years to come. I wanted to mention also a couple of observations and advancements that have been made in imaging. And this, I think what's exciting about this is I think we've known for a long time that we can take great pictures of the lung. CT is really gives an absolutely beautiful picture of the lung. But there's been some recent advances in MRI which I think might make it an attractive option in the future. What we're looking at here is a patient, a young patient with CF, I think about 2 years old, who had both a CT and an MRI performed at the same time. And the MRI is slightly different than the usual. Historically, MRIs don't take very good pictures of lung. They just don't deal well with too much air. And there's been some advances in how that data is captured, called UTE, or ultra-short echo time, which basically allows us to get a structural image that's not that different than a CT. And as we look down in this, we can see that there are many of the features that are characteristic of CF lung disease can be captured by both modalities. What is exciting about this is that using these imaging modalities allows us to look at regions as opposed to the whole lung. Most of the measures that we use today, such as pulmonary function tests, lung clearance index, which is in development, they're global and they're great, but they're limited because they're global. And what imaging allows you to do is look in places that may be where the source of problems are. MRI also, though, has an interesting sort of side to it that you can look at function. You can look at blood flow going through the lungs, and you can look at air going in and out of the lungs in different regions. And that also provides us just a really beautiful and elegant way to look at interventions. Finally, I think, at least in principle, this could become a lifelong tool. Right now, we don't have any tools to monitor CF patients that begin in the first year of life that we can take through their entire lifespan. I think this at least offers that possibility, and we look forward to how these will be developing over the next couple of years. Just to show an example of functional imaging, this was published earlier this year. The top is showing structure of the lung, and this is from a patient before he started antibiotics and after for a pulmonary exacerbation. And the arrows, I know it's hard to see, but what it's picking up is these kind of subtle abnormalities that get better. And so what it appears is that the MRI has the sensitivity to pick up minor changes and also detecting their improvement by an intervention such as treating a pulmonary exacerbation. Down below is just an example of a perfusion scan of a patient, same patient, before and after treatment of a pulmonary exacerbation. And what they're showing is a defect in perfusion in the top right lung. Sorry about that. Yep, your left, patient's right. And that sort of dark spot, and that really is a place that likely represents a mucus-plugged area and doesn't get normal perfusion. And on the right side, as the patient has been treated, that place now has normalized perfusion. In addition to that, evidence or how can we look at the lungs in terms of where the air goes? This is an example from Jason Woods and Zach Cleveland, where I work. They're just down the hall. The top is a picture of the lungs using this UTE MRI modality. The middle is the same person breathing in a hyperpolarized gas, which basically tells us where air is going, and importantly, just as important, it tells us where air is not going. And then the bottom image is putting those two things together, the structure shown from above and the function of where the air is able to go. And what you see is that top picture kind of missed some of those areas where ventilation is not happening. And so I think this type of tool is going to be an incredibly valuable way for us to optimize therapies for patients in the future. And so there are a number of exciting novel biomarkers that are under development, including going for CFTR bioactivity, looking for sensitive biomarkers of early drug response, biomarkers of lung remodeling that will help us identify disease modification, and then finally peripheral biomarkers which will help us to monitor disease. And some of these, we hope, will become what are called surrogates. And surrogates are basically predictors. They're the types of things that are going to tell us whether they translate into what are called clinical efficacy measures. And basically these are measurements that the FDA feels confident to help us know that that patient is going to feel better, function better, or survive better. And so this will be bringing these different types of analyses into clinical trials today is really going to help us accelerate both making treatment decisions and also accelerating drug development. All right, quick question. Who's got a Fitbit on? Who should have a Fitbit on? All right. So what is Fitbit? Obviously, it's a personalized monitoring tool, and I have one on my phone, and as I'm walking around, it reminds me how many steps I haven't taken today. And I think the point of this is, though, that there are apps in development that we're using every day that you could see could become means to track symptoms in CF patients. These can track activities, they can track symptoms, they can track therapies. And what we'd love to see is this type of app that is able to interface with the electronic medical record, and it's going to help us in a lot of different ways. It helps us with pre-visit planning, making a care plan while in clinic, and monitoring interventions with a lot denser data rather than just, well, how you've been the last 3 months. You've actually come armed with, well, I can tell you Tuesday my cough was really bad, but by Wednesday it was sort of not so bad, and maybe there's a little much, but you know what I mean. And one platform that's being targeted, or excuse me, that's being developed or at least tested where I'm working is called Orchestra. And I did want to do a shout out that there will be a CF Model Design Project Innovation Reception that sort of looks at some of these capabilities that will be on Friday night at 8 o'clock over at the Sheraton. So if you want to see some of this cool stuff, I know it's being shown there. And why is this important? Well, there's a recent study that was published by Syracuse and colleagues that looked at the use of ivacaftor in patients who were prescribed using a variety of different monitoring tools. This included 6 patients who were pediatrics and 6 adults, and they'd been on drug for over a year. So this was not like just when they started, but this was over just kind of picking up after they'd been on therapy for a fairly long period of time. So 11 of 12 had a self-report of 100% use. If you looked at pharmacy refill history, something called medication possession ratio, or MPR, that was about 84%. But if you actually looked at electronic monitoring, where they had little caps or little monitors that would pick up when caps were open and closed, that's about 60%. And that's what's shown on the top is basically looking at the weekly electronic monitoring of medication over time and for each of the patients. And I think the point of it, there's a lot of lines and they're doing this. And the point is there's probably not a one-size-fits-all approach here. The bottom is showing the dosing interval. And what I took home from that is we know that ivacaftor works best if taken every 12 hours, really based on its pharmacokinetics. The average dosing interval was about 16.9 or 17 hours. So there was long periods between doses at times, and it also varied quite a bit from patient to patient on a week-to-week basis. So to tackle this type of question or these types of challenges, the CFF is supporting a new work group, the Success with Therapies Research Consortium, and their focus will include adherence and self-management. It's being led in part by Cindy George and other people in this audience, and they'll be really seeking to optimize outcomes and quality of life for CF patients. And I look forward to having a plenary coming up in the next year or two showing us how that has really impacted things. So how can apps potentially drive engagement? Well, there's a few things they need to be. They need to be scalable, which basically means that we can use them a little or use them a lot. They need to have sort of thoughts about what works for a person, sort of a behavioral type of design. And we're hopeful that this would lead to more engagement, collaboration between the families and the care team to optimize outcomes. This can allow things like symptom observation and monitoring, but it also can help patients learn more about their disease and also, at least in theory, to monitor what they're doing at home. And this is just a very simplified example of that was shared with me by a CF parent. This patient her child has problems with a multi-resistant bacterial infection, and she cycles 14 days on and off an antibiotic to try to manage that. And what we're seeing as we move from left to right is the time is basically in days. The top is basically picking up whether they're on therapy, which is above the line, or the lower line is when they're off therapy. And the bottom is monitoring one symptom, and they're just monitoring the cough. And what you can see is when on therapy, there's not much cough. When they go off therapy, cough starts to go up, and then when they go back on therapy, cough starts to go back down. And while this is a relatively simple example, I think you can see how this type of monitoring tool could be really valuable to a given patient and family. So I hope what I've been able to show you today is perhaps we're not too far, and perhaps we're even right on the cusp of taking a patient's or a patient's cells and trying to make decisions about what might be a good option for them. So starting from the left, we have an undefined mutation, or basically maybe a common mutation that has many therapeutic options. We then go to the patient, a patient-derived model system that helps us look at whether these interventions or these different kind of options seem to make most sense for them. Take that model system theoretically into a novel personalized trial, and perhaps we now have a way to really have the most optimal therapy that's appropriate for a given patient. So as I'm just getting ready to close, I know I'm standing between you and I think the reception, so it's time to get a move on. But first of all, I think it's safe to say that personalization with precision is really, I think, one of the most exciting, if not the most exciting, next step in CF care. Genotypes and theratypes, they're emerging ways for us to consider these new therapies. Patient-derived model systems, they may be a valuable platform for us to predict benefit, but there's obviously work to go on that front. Novel biomarkers will help us accelerate trials and hopefully streamline therapies, help us know when it's safe to not use a certain therapy, and I think that's just as important as adding something, obviously. Addressing adherence and engaging the patients on a personal level is really critical if you want to have the best and improved outcomes. But I think we're on the cusp of harnessing the data and experience of patients, and that's going to allow us to have really a truly personalized healthcare system. So I just wanted to read quickly. Bob opened up with a note from a patient who had initiated a modulator treatment, and I have one myself that was sent to me as an email. And I just found it kind of inspirational as I think about all the clinical trial work that we do. Sometimes we forget, that what it means to the patients who are participating. And it's also for me sort of a segue to tomorrow's plenary, in which George Retch Bogart's going to be talking about the importance of clinical trials in CF today. So this is from this guy. His name, he will remain nameless though. This week I received my first official prescription of commercial Lumacaftor Ivacaftor. I know the drug development process is a long one, of which I am only a small part. I know there are many hoops to jump through, boxes to check, I's to dot, T's to cross, and on occasion, go have fun with friends. Additionally, I'm certain there are at times this feels like work. But you need to hear someone say thank you. Although all the paperwork and data may feel like another day at the office, this is work to get a drug in my hands that will and has improved the quality of life and added years to my life. You are doing important jobs that affect a lot of people, and you deserve thanks. So thank you for all you've done and are continuing to do. I will continue to serve in whatever roles I can in the development of future therapies, but you all are the workhorses. Keep fighting the good fight, and I'll look forward to hearing about the next trial. And that to me is just so meaningful that we made this great accomplishment, and he's already looking what's next, and that really speaks to me what the community has to say. So I wanted to say thank you very much for your attention. I know it's been a long plenary. It's not my fault. No, I'm kidding. And I wanted to leave you with a really meaningful video that was shared with me. And if you take it there.
Get happy.
It might seem crazy what I'm about to say. Sunshine, she's here. You can take a break. I'm a hot air balloon that could go to space. With the air, like I don't care, baby, by the way. Because I'm happy. Clap along if you feel like a room without a roof. Because I'm happy. Clap along if you feel like happiness is the truth. Because I'm happy. Clap along if you know what happiness is to you. Because I'm happy. Clap along if you feel like that's what you want to do. Here come bad news talking this and that. Probably warn you, I'll be just fine. No offense to you, don't waste your time. Here's why. Because I'm happy. Clap along if you know what happiness is to you.
So go out there and have fun, all right? Thank you again.